Hi guys, this is Dr. Gonzalez here, and in this video I'm going to talk to you about replication and credibility in research. And specifically, we're going to talk about how certain cases have happened and how they have contributed to a replication crisis, and some other examples of how credibility of certain researchers has completely been demolished because of their poor choices. So let's talk about replication and credibility. So what is replication? Obviously, to replicate. Replication is the process of repeating a study again and again to see if the same results will occur. And it's very essential in the scientific process because one single study can't prove that a particular finding is true. So we need to go out and do replication studies to provide more credibility to our results. Why is replication important? Well, replication has three particular things that it increases. Number one, it increases our confidence in the results that have already been obtained. Two, it helps bolster the credibility of the researchers. And three, it increases the reliability of conclusions that we've already gathered. So this is all coming from what we call the replication crisis. This happened in the early 2000s, and it refers to this general concern that came out when it was found that some published findings could not be reproduced by other researchers. And it ran through several different fields of science, including psychology, medicine, biology, neuroscience, and economics, just to name a few. Around 2005, someone named John Ioannidis published an article called Why Most Published Research Findings Are False. And in it, he argued some particular points. Number one, he said a lot of findings are likely influenced by using small samples. So we've discussed the importance of large samples in the past and how the sample size is related to aspects of validity. Well, what he found was that a lot of published research is based on extremely small sample sizes, or findings that are based on small samples. He also postulated the existence of something called publication bias. And what that means is in the publication world, there tends to be a bias towards only publishing positive results. So if a study is conducted and it has a different result from the positive, that study is generally not published, even though it may lend credibility to something that we are looking at in science. He also talked about how many studies have really weak statistical power and how we generally over-rely on statistical significance when we're talking about our results. So you've talked with our hypothesis testing about how a hypothesis test can be done and we can fail to reject or we can reject a null. And what this is saying is that sometimes when we reject a null, the results are determined to be statistically significant, but they may not be relevant or applicable to the general body of knowledge. So when this replication crisis came out, it continued to grow, and there were some major concerns in the 2010s in psychology. And the major concern from all of this is that our research system, the publication system, it rewards findings that are new or surprising or statistically significant. And it doesn't put as much emphasis on studies that are carefully replicated. So in other words, if it's new, exciting, surprising, and significant, it kind of trumps a duplication or a replication study that could have also contributed to whatever it is that we're looking at. So with the replication crisis, you've got to remember that researchers sometimes have an incredible amount of leeway and power in their work. They can choose which variables to study and then which variables to report. They can also exclude participants from studies. So maybe there are participants out there that might not have supported their conclusion if their data were included. So it's completely acceptable for people to exclude those participants. Sometimes we stop data collection when we've got results that are significant. We don't continue out with a study. And the big one to me is that sometimes we do many analyses, but we only report the ones that are significant, and we kind of ignore the ones that weren't. The crisis kind of came to a head in 2015 when a group called the Open Science Collaboration published the Reproducibility Project in Psychology. And what they tried to do was to replicate 100 psychology studies from major journals. And a lot of these studies were big studies, important studies that you would think would be worthy of replicating. And if the results supported the original conclusion, it would lend more credibility and trust to the scientific community. Well, what they actually discovered was that a lot of replication attempts produced weaker effects than the original study, and when it was all said and done, only a small portion of the original findings could even be replicated. So that shows that we've got some problems out there that we need to be wary of, and that we need to be conscious of these issues when it comes to publications. Some of the problems that they came up with in the study of this replication crisis included studies that had really small sample sizes or very low statistical power. They also found the issue of publication bias, the thing that we talked about earlier, where only studies that are super new or surprising are published, and we kind of ignore things that are not. They also talked about p-hacking and harking, which we will talk more about in depth in this video later on. It also discovered that a lot of times the methodology sections of these journal articles are vague, and that is a contrary point when it comes to replicating studies. If you're going to replicate someone's work, you really need to know what their methodology and their specific procedures were in order to truly replicate what they did. There's also this general hesitance in sharing data, especially the raw data that's out there. So after a researcher conducts their study, they hold on to that data, and there is not this like central repository where people can get the data and replicate the statistical analyses that are being conducted. And then finally, there's just a general lack of studies that are replication in nature. So there's been some work in response to this to establish some reform and to effect some changes in the research world. And some of these changes include some items that we're going to talk about later on in this video in more depth. But some of these reforms and changes are things such as pre-registration of your hypotheses, pre-registered reports, using larger sample sizes, making our data and our data sets open to other researchers and even to the public, having more open materials, including the methodology and procedures that we're using in our studies. There's also been a general emphasis on more direct replication studies, and also a little bit more emphasis on effect size and confidence intervals when it comes to our statistics. So it's not just about that p-value related to whatever statistic we're talking about. It's also the p-value, the effect size, and the confidence interval for our particular results. So why are replication studies important? There are several reasons. So first of all, we just need to remember that a single study does not prove anything. We can't just do one research study and make a conclusion from that and that be the end of everything. We need to have replication studies, and there are some really important reasons for that. So first of all, replication studies helps us as researchers and scientists to confirm previous findings. So when we do these replication studies, it provides a confirmation that the findings that were already found were real. And by replicating them, we demonstrate that that study can be repeated. And by repeating it, it helps us determine if those original findings were accurate or was it just a fluke or a chance that we found those results to begin with. Next, replication helps us to increase scientific credibility. So your credibility as a researcher is bolstered by replication studies. So if there are multiple other researchers out there that come up with the same results that you did, the trust in your results and in you will be increased. And so when other researchers and then finally the general public have more trust in a conclusion, it's going to be found by demonstrating the results multiple times instead of just the once. Replication studies are also important because they help us identify errors or bias. We have to remember that there are always possibilities that a mistake was actually out there, right? We committed a mistake either on purpose or inadvertently, right? We can also have flawed assumptions in the design of our method or our methodology. And then we always have to fight with the idea that bias exists. Replication will help us increase the probability of finding a mistake, a design flaw, or some sort of bias. So let's talk about other reasons why we need to replicate. So replication studies are important because first, they test reliability across different samples. We talked in many of our previous videos about external validity and how we want to make sure that our results are valid by looking at those results in other populations, other settings, other locations, and other times. So by repeating these studies with different participants or in a different location or at a different point in time, it helps us determine our external validity, and it helps us to apply the results to a wider population. Replication studies also help us to reduce the impact of a false or an exaggerated findings. So if a particular research protocol is being used multiple times, we might have one weak result in there. And if we've repeated it multiple times, that weak result is not going to hold as much weight. So replication corrects the idea that a strong effect was found that later on someone else determined that effect to be weaker or even non-existent. By replicating it, we can correct that. And also, replication of research studies helps us strengthen our theories and our general body of knowledge about a scientific topic. So when you see consistent results across many studies, it proves that our theory is more likely to be true, and it provides a stronger support for that theory in the scientific community. Let's talk about the different types of replication. The first kind of replication is called direct replication. And in direct replication, we are going to repeat a study as closely as possible to the original conditions that study was conducted in. So our goal in this particular type of replication is to verify original results. So if our results are similar, it provides stronger evidence for the original results that were found. So here's an example, and this example is going to be used throughout my examples of replication. But in direct replication, we would have a situation like this. A researcher conducts a test looking at retrieval practice or quizzing, and we want to know if retrieval practice improves memory compared to just rereading your notes. So you've got two groups here. One is doing retrieval practice through quizzing, and the other one is looking at their notes, just looking at their notes. In the original study, the results showed that students who took practice quizzes did better on a final test than participants who just read their notes. So if we were going to do a direct replication in this particular situation, you're going to have a new researcher using the same procedures. So it's going to be the same quiz, same timing, and the same test materials. And they're going to try to follow the same steps as closely as possible that were used by the original researcher. And they're also going to use a similar group of participants. We're not going to move into a new population just yet, because our goal is to directly replicate the original research. So if our study finds the same results, obviously this helps support those original findings. The second kind of replication is called conceptual replication. And in this version of replication, we're going to test the same idea, but we're going to use different methods or measures. And our goal is to show that our findings are not specifically dependent on one specific procedure. So using the same situation we talked about in the previous example, we're going to talk about a researcher studying whether retrieval practice improves memory as opposed to rereading notes, right? So the original study, they either read their notes or they took some short quizzes. And we found that those who took the quizzes did better. In a conceptual replication, the researcher is going to take that same idea, but they're going to change the method. So this time, instead of using maybe short answer quizzes, we might use multiple choice quizzes. And this time we also might want to change the material that they're being tested on. So this time we might use vocabulary instead of reading comprehension passages. But the goal of this is to see the same pattern of results, even if the procedures are different. The third kind of replication is called replication plus extension. And this is where we repeat a study and add new variables or conditions to it. And our goal here is to expand our knowledge and to test new questions related to the original. So with our same situation here, a replication plus extension study might be when a new researcher repeats the original study and they add a new condition to extend the research. So this time we might add a third group using flashcards or spaced practice. Or we could compare blocked versus interleaved quiz. And by doing that, we've now extended the original research. And we're going to compare our findings to the original conditions. And we're going to see what we find. Now, large replication projects are important, and this is when we have multiple research teams that are conducting the same repetition across many different locations. So each team or location is going to use the same standardized procedures, and that allows researchers to test if our results are going to be consistent across different populations, different settings, and different researchers. So when we do this, our projects are going to be designed to produce an outcome that is more reliable and findings that are more generalizable than just a single study. Now, these large replication projects allow us to do or find several different things. First of all, they test consistency of results. So in other words, if our findings are true across many different labs, then our findings are more likely to be valid. Large replication projects also reveal variability. So if the results differ based on location or sample or context, we know something else is going on, and that warrants even further research, maybe even more replication. These large replication projects also provide accurate effect sizes. So when we increase our samples and our sample size, that helps us demonstrate that our effects that were originally found are going to be larger because we're using a larger sample. And then finally, replication studies on a large scale increase transparency and collaboration. So anytime researchers are sharing their data, their methods, and their results more openly through collaboration and transparency, they're helping to improve and to continue to build trust. Now, meta-analysis is a very important statistical method that you need to keep in mind. Meta-analysis combines the results from several different studies and it produces an average effect size across research. So what it basically does is it helps us determine the overall strength of all of the evidence out there for a particular research idea. Now, it has strengths and limitations. Its strengths are it uses large data sets, and the larger the data, the more reliable our conclusions are generally going to be. It also helps us identify patterns across studies. But the limitations are it can be biased if it only uses published studies right? So we have this idea of publication bias. We're only publishing results that are statistically significant. And since we've only got access to those studies that are statistically significant, your meta-analysis is only as good as the previous studies that have been conducted. Okay, and then also the quality of your meta-analysis depends on which studies you included and their quality. So in a meta-analysis, researchers are going to collect data on many studies about the same research question. Then they extract the effect size from each study and combine them into an overall result. And obviously, like I told you previously, studies that have larger sample sizes are going to get more weight because the larger the sample size, the more reliable the results are. So let's talk about effect size. We talked about this when we talked about our specific statistics. We talked about correlation and regression and our chi-square. Those all come with an associated effect size, right? So effect size measures the strength of that relationship or the difference. So it goes beyond just saying this was significant or not. It talks about if it was significant, how big the effect was. So when we have a small effect size, there was little difference between our groups. When we have a large effect size, there is a larger and more meaningful difference. So in a meta-analysis, we come up with more reliable conclusions because we have now combined data from lots of different studies. By doing that, we also increase our sample size, and the larger our sample size, the greater our statistical power and our accuracy. Remember, meta-analyses help us define and decide on patterns, and that helps us determine when and where those effects actually took place. And meta-analyses are good for resolving conflicting findings. So a meta-analysis might help us explain why some studies came up with different results than others. There are limitations, like we said, there's the publication bias. We tend to rely on studies that were only significant to get published. A meta-analysis is also subject to garbage in, garbage out. And by that we mean if we include poor quality studies in our meta-analysis, your results that you get are also possibly going to be misleading. And if there are differences in methods or samples or measures, it makes the comparison across studies more difficult. So there are inherent differences in the studies that may not lend themselves to a good meta-analysis. So why should we worry about this? Well, meta-analysis gives an opportunity for us as researchers to look at a bigger picture of the research findings. It also helps a scientist to make a more evidence-based conclusion, and it does play a role in psychology as well as medicine and education. Let's talk about some questionable research practices. There are lots of different questionable research practices, but by and large, the three most common ones are p-hacking, harking, and underreporting of null results. So we're going to talk about what these three things are and go into a little bit more of a deeper conversation about them. But the bottom line is that each one of these questionable practices reduces trust and the ability to replicate studies. P-hacking is when we basically hack what is considered to be a statistically significant result. So a researcher goes back and they manipulate the data analysis until they get something that was statistically significant. So typically these researchers plan one kind of analysis, right? And if they conduct that analysis, and as you see over here in this picture, they do analysis number one and they get an insignificant result, right? A result that's not significant, they continue to do more analyses until bam, finally we get this result that is significant, and that's what gets published. So all of these other comparisons or statistical tests that they ran, they were not significant and they basically just get thrown out. And this is just, it's not a good procedure to follow as a researcher. So what might happen is your researcher tests multiple variables, but they only report the ones that were significant. They may also try different statistical tests until they finally get one that gives them the desired result. they may go in and actually start modifying the data. They may remove outliers until they get results that are significant. And we talked about how early termination of research studies once they've reached significance happens. So what that means is a researcher might stop collecting data once they've got a significant result. And then they may also take a large data set and start splitting it into subgroups until they get a significant effect in one of those subgroups. So generally these choices are made after the data have been collected and they're not planned in advance. So in other words, the data, the procedure, or something has been manipulated afterwards to contribute to a significant result. Now, p-hacking is a problem because, number one, it increases false positives. So the more you go looking and fishing out there for a significant result, the more likely you are to find one just by chance. Okay, so you may find something that is significant, and you may be very happy about that, but the ability to find that significant results might be just due to the probability of finding a result. P-hacking also reduces the replicability of a study. So if a study produces results through p-hacking and someone comes behind them and tries to replicate it, they may fail in that replication study because they're not going through that fishing expedition to find those significant results. And then p-hacking in general can lead to misleading science and the public because it creates this kind of illusion of strong evidence when in actuality there was little to no evidence to support the research question that was being postulated. So here's an example of p-hacking. So say you have a researcher who wants to test whether music improves test scores, and they try different genres of music. They go through jazz, they go through rock, they go through classical, they go through heavy metal until they find something that's significant. Or they try different time lengths. Maybe they start small or large and go in the opposite direction until they find something that is significant. Or maybe they just test it on multiple different groups until they find a group that happens to be significant. So you can see where the possibility of finding a result is going to increase with the more groups or variables you're exploring. So after they do all of these multiple different tests, they do get a significant result, and that's the only one that they report. The actuality behind this is that the significance that they reported is probably just random and it's not real. Now let's talk about HARKing. HARKing is an acronym that stands for hypothesizing after the results are known. So this happens when a researcher creates or changes their hypothesis after they find the data or use the data. And then they actually present that as what they had planned to do all along. So in general, we're going to have this idea about what our research protocol is going to be. We're going to set a p-value beforehand, and that's great. But HARKing is when we do that. We establish our p-value, our probability, our alpha level, right? We collect our data, we analyze it, and then we find that our results were not significant. So instead of going back and redoing the experiment or collecting our data again, we just change the hypothesis, okay? And we present that hypothesis as actually being what we thought was going to happen all along. So researcher runs a study, they may not have a clear prediction, or their original hypothesis is not supported. After they look at the data, they see something interesting. So then they rewrite their hypothesis to match the results, okay? And they're going to go and publish their results as if their hypothesis came before the data. So in other words, they've kind of got this sneak peek at the data, and then they write their hypothesis. And then they pass it off as if they had written the hypothesis from the beginning. HARKing is a problem because, number one, it misrepresents the scientific process. If you go back and look at the hypothesis testing procedures, our alpha level or our significance is set before we do the statistic, okay? Second, it also inflates confidence, okay? If our results are going to seem stronger and more reliable than they actually are because we've now crafted our hypothesis to fit the data. And again, it does just like p-hacking in replication in that it makes it more difficult. Since you're basing your findings on what's called post hoc or after the fact reasoning, it's going to be much less likely that someone else is going to be able to replicate your study. So here's an example. A researcher studies whether sleep improves test performance. They do their research and they collect their data and they find that there was no effect of sleep on test performance, but they did find a relationship between stress and the test performance. So instead of reporting honestly that they didn't find a relationship between sleep and test performance, they paint a picture of, well, we were studying stress and we found that stress affects this, and that's what we predicted from the beginning. So they changed their story after they saw their results. The third thing that we need to discuss here is underreporting null results. So underreporting null results happens when we don't publish or we don't fully report studies that didn't find a significant effect. So a null means the study didn't have a meaningful relationship or a meaningful difference between variables. So when you underreport it, you're, not reporting things that were not as exciting or not as interesting. And that happens because journals tend to prefer exciting or significant results. There's also a pressure for researchers to publish, and that correlates to a pressure to publish positive findings and avoid things that might look like a failure. And then in general, null findings are not necessarily valued as much as significant findings, and they're often seen as less interesting. Now, underreporting null results is a problem because, number one, it only paints a particular picture of our scientific record. In other words, literature ends up showing the positive results, even if there were 5 times as many studies that showed no results whatsoever or no effect. So in other words, we're focusing on the positive results when maybe there's only 5 studies that produced positive results, but maybe there were 50 that found no effect. So we're creating a biased scientific record. It also overestimates our effect sizes. It can make a relationship appear to be stronger than it actually was. It also misleads future research and decision making. So if you've got scientists, educators, and policymakers out there that are relying on our research and they're only being given an incomplete picture with the significant results, we're painting an inaccurate picture for them, and they're basing their decisions on incomplete or inaccurate evidence. And then finally, underreporting null results continues to contribute to the replication crisis. So when we don't publish our null results, when we ignore those, findings that are published are going to be less likely to hold up over time. Okay? Here's an example. So maybe we had 0 studies test whether, I should say 10 studies. 10 studies test, I'm going to put a 1 there. 10 studies test whether a new teaching method improves test scores. 7 of them had null or no effect, and 3 showed a small positive effect. We only published those 3 studies, and so it may look like the methodology worked, even though a larger body of evidence, 7 studies, showed otherwise. So how can we improve research? There's a lot of different ways, and there are a lot of new initiatives out there that researchers are coming to terms with. We want honesty in research. And so some of the ways we can achieve that honesty is through having open data, open materials, and pre-registration. Okay? Open data means we're going to share our data with the public. So that way someone else can come along and they can replicate our data analysis, and they can back us up basically with our statistical analysis of our data. We can also share our materials, which is the methods and procedures that we're using. We don't hold on to them as being proprietary, and we share those so that other people can replicate our studies, because by replicating them, we're actually letting them help show that we were correct, right? And then pre-registration is something else that is happening out there. This is where researchers are coming forward and stating their hypotheses and methods before they start collecting data. So they're basically sharing what they're going to do and what they think is going to happen beforehand, and that helps kind of hold them accountable when they publish their results. But these solutions here help us to increase honesty, accountability, and reproducibility across our research. Remember, we're always concerned with external validity, which is generalization. We want our external validity to be high as much as we can with respect to our internal validity. But external validity is how well our results apply to other populations, times, or settings. And we're really concerned with whether or not our sample was representative of our population. So you have to remember that research is all about the population, not just the sample, okay? What you found in your sample has to be true of the population. You've got to remember that random sampling is always better than convenient sampling. We use convenient sampling because it's convenient, and there are times when the only group we're going to have available to us is a group that is convenient. It's not always possible to do random sampling, but in the end, random sampling trumps convenient sampling. And you also have to remember that a bigger sample is usually good, but it does not equal better if that sample is biased. We also have to take into consideration who is being studied. Most research in psychology uses convenient samples. Those samples are often pulled from college students, and they're often what we call WEIRD, which is an acronym that stands for Western Educated Industrialized Populations. Because we're limited to these Western Educated Industrialized college students who came to us through a convenient sample, our results are going to be limited in their application on a global scale. You also have to consider the different cultural aspects. Certain psychological processes are not the same across cultures, so as a researcher, you have to be aware of that and avoid that assuming that your research findings are going to be universal and the same across different cultural beliefs. So the key takeaways I'd like you to remember from this is that replication is very essential for trustworthy science. A lot of the findings that are out there are now being seen as less reliable than they once were. We can improve research by including transparency, be mindful of generalization, and as a discipline, psychology has to move beyond those Western educated industrialized samples. Now let's talk about some researcher misconduct. There is an assignment for you to do related to this topic where you're going to read about someone who I'm going to mention in this video who conducted some misconduct. But let's talk about some examples of this. So the first one is a guy named Diederik Stapel. He was a social psychologist and he got in trouble for fabricating data. He's a Dutch psychologist. He had published in some major journals, and it came out that he had fabricated or manipulated his data across dozens of his publications. And what ended up happening was these publications, including this example right here, Coping with Chaos, How Disordered Contexts Promote Stereotyping and Discrimination, it was retracted. So because he did not analyze his data correctly, because he made up data, and because he altered his data, this is an example of someone who conducted data fabrication, and it came back to really, really haunt him. An example here from the United States is Mark Hauser. He was a psychologist who worked with animal cognition, I believe at Harvard. He was an evolutionary biologist. He was studying how primates think, primate cognition. It came out that he had also fabricated data, he had manipulated his results, and he had reported findings that were false. And to make matters worse, when things came to light, Harvard conducted an investigation, and Harvard was not truthful about that probe, right? So other researchers said, hey, we get that these things are happening, but when an institution like Harvard is made aware of this and they don't share that this allegation is going on, that's almost as bad as whatever Dr. Hauser did. So there's accountability not only to the researcher but to their institution as well. Huang Wusuk was a stem cell researcher who got in trouble for fabrication of some stem cell data. He claimed that he had created an embryonic stem cell line through cloning, and that ended up being false. And those publications were retracted. I basically just included this because it was an example of a hard scientist, someone who was in the biological sciences. So this isn't just happening in psychology, it's also happening in the hard sciences like biology. The researcher you're going to read about in your assignment is Brian Wansink. He was a food psychology and nutrition behavioral researcher. So he actually wrote books about our eating behaviors, why we eat more than we think. He did a lot of research with buffets and having people estimate how much they ate when they used like a small plate versus a larger plate. And he was popular. It wasn't just that his research was being conducted in his research world. The media found out about it. He was writing these books, and he became popular for his theories in other areas other than just the scientific community. But later on, they found out that his papers were retracted or his data techniques were questioned because he basically involved himself with p-hacking and data dredging. So he had these massive databases from these studies that he had conducted, and he basically told his graduate students to go out there and go fishing or do what's called data dredging until they found results that were significant and could be published. And you'll read more about him in your assignment for this week. And the one that I want to speak about the most, because for me it was very impactful, was Andrew Wakefield. He may be one of the most famous examples of the danger that data fabrication and p-hacking and harking and all of those things can be to not only the research community, but to the general population. So Andrew Wakefield was a UK physician. He published an article in The Lancet in 1998, and that article suggested there was a link between the measles, mumps, rubella, or MMR vaccine, bowel disease, and autism. This was the first researcher who kind of put on the scene the idea that there may be a relationship between vaccinations and autism. The paper was retracted. Unfortunately, you can see here it was retracted 12 years later. Wasn't soon enough, in my opinion. And Wakefield was also stripped of his medical degree because it was found that he was very dishonest and irresponsible. He treated his research subjects who were children unethically, and he did not disclose some significant conflicts of interest. The most surprising one to me is that he actually had his own vaccine that he had patented and wanted to sell to replace the MMR. So it was in his interest to show, even if it wasn't a true link, it was in his interest to put a link out there, because even if it wasn't true, he was going to benefit from it. And for me, this is a very strong example of how dangerous scientists can be, because people are still believing misinformation that was started by Wakefield in 1998. So what was wrong with his research? The publication in The Lancet had a sample size of 12 children. And these 12 children had both a developmental disorder and some tummy trouble. They had gastrointestinal disease or symptoms of gastrointestinal disease. So he wrote this paper and postulated that there was a connection between the MMR vaccine, intestinal inflammation, and that inflammation then caused autism or pervasive developmental disorder. Now, his paper did not prove causation, but he completely latched on this idea that there was a causative pathway here, and he promoted that idea that vaccines caused autism. And his argument was that these autism-like symptoms started after they got the MMR, and that the MMR actually caused a condition in the bowel called autistic enterocolitis. So in other words, you took the MMR stimulated something to go wrong in your bowel, causing this enterocolitis, and then the symptoms of autism followed that. Problems with it continued. Again, there were only 12 children in the study, and 8 of the 12 children had symptoms. So not even all of the children had symptoms. The big problem that you can see also from here is that it's a very small sample. Okay, 12 participants. You can't make a claim, even correlation with that. This was a case series. That means that it wasn't even an epidemiological or an experimental type study. And we get that. We can't always do experimental or even epidemiological studies on a grand scale. But he did not even include a proper control group. You couldn't determine if the symptoms were even more or less common in other groups of children. So in other words, if we had a sample of children with autism that did not receive the MMR and we looked at them, we might have been able to discover something else out there. But because there was no comparison group to children without autism, and there was no comparison group of unvaccinated children, we couldn't do this. If we had taken a group of unvaccinated children and looked at their rates of autism and this autistic enterocolitis, we might have been able to establish more of a correlation, but we couldn't do that because the study was based on 12 kids, and that was it. So some other problems with the study, it relied heavily on parental reporting of when the symptoms began. And we know that when we ask parents to recall information from many years ago, it can be inaccurate, especially when we ask them to reconstruct a timeline of these complex developmental changes, right? Later investigations also raised concerns that this symptom onset timing had been misrepresented or manipulated, outright manipulated. And then some of these children were actually partners to ongoing litigation against vaccinations, actually vaccination manufacturers. So what he did was he recruited these parents and families who were suing the vaccine manufacturer. So they already had a sort of inherent bias against those manufacturers. So sure, they're probably going to report a timeline that supports the idea that these vaccines did damage. And then here's the one for me that just takes the cake. Wakefield had a patent for a rival MMR vaccine. So in other words, he was coming up with his own vaccine against measles. Actually, it was just a measles vaccine, but it did rival the MMR vaccine. And he had also been paid by lawyers that were involved in litigation against vaccine manufacturers. So the people who were funding his study were actually lawyers who were suing vaccine manufacturers. Those are clear conflicts of interest, and those things have to be disclosed. Here's some other things you might not have realized if you've read about this. Some of these kids in this particular study went through some very invasive medical procedures. They had colonoscopies done on them. They had lumbar punctures and spinal taps and other invasive medical testing. And that brings up an ethical concern, because if you're doing a study here to look at an association claim between vaccines and autism, there are significant ethical concerns that these invasive tests were not justified and they did not have any sort of ethical approval. His data was also manipulated and misrepresented, and that included the histories of the children's medical symptoms, including the onset of their symptoms with the timing, the findings of those bowel studies. He had no ethics approval, and he didn't disclose those conflicts of interest. So there was also a lack of scientific literature supporting his claims. There were studies that were conducted later on that failed to find this supposed link that he was postulating. The co-authors on his actual publication in The Lancet withdrew their support in 2004, and then The Lancet actually said, we retract this article in 2010. So it took 20 years. No, it took 12 years, but it did come to light. So the main takeaway from this is that his paper was not strong evidence that the MMR causes autism. It was a super small, uncontrolled case series, and it had significant design flaws, ethical problems, conflicts of interest, and then dishonesty. But it's an important case to look at because despite being a weak and flawed study, it had significant impact on public health because it raised fear about vaccinations and reduced the public's trust in vaccinations. So we're left with these consequences 30 years later. That's the end of our presentation for this. There is a short assignment that you'll have to do that will be published in Canvas. And if you have any questions, please reach out and contact me. I'll be happy to answer them. Take care.